Hello, everyone, and welcome to our free webinar. We're going to be starting here in just a minute, so hopefully everyone can join on time. Good morning, and welcome to this free webinar. In this session, we will discuss our upcoming training program called the Omicslogic Data Science and the online research opportunities for students, faculty, and industry professionals that you can leverage to apply bioinformatics to complex and fascinating data sets, demonstrating your data science skills and learning about biology, different diseases, and the treatments that are coming out from the data that we have about these biological conditions. So I'm excited to welcome you to this free webinar. Again, we're just going to start here in just a couple of more minutes as everybody is just joining right now. And I'm looking forward to also interacting with you and getting to know all of you, hoping that you will attend the full program that will start on August 3rd. Now, I know that for some of you, this is not morning, this is actually evening. So we're here in New Orleans and it's morning time for us. And this recording of this webinar is also going to be available for future reference. Okay, so for those joining us for the first time, my name is Ilya Brodsky. I'm the co-founder and CEO of Pine Biotech. Our vision here at Pine Biotech is to enhance human health and well-being by enabling biological research and discovery with relevant data, solutions, and expert support. Our mission is to simplify bioinformatics and advance research through our modular and intuitive multi-omics analysis platform powered by human experience and artificial intelligence. You can read more about us at pine-biotech.com. But what our company does is we offer training, research support, and big data analytics tools designed for research and biomedical discovery. Our mission is to support cutting-edge research projects and solve big data challenges by developing innovative solutions that are easy to use and to provide training to use them effectively and help scientists arrive at sound and reproducible results. I'm honored to have been working with a team that collectively have an amazing amount of experience in this area, including the other co-founders of the company, Dr. Alfred Tauber and Dr. Leonid Brodsky. They come from a medical background with expertise in oncology, immunology, and biochemistry of inflammation, as well as bioinformatics and biostatistics. Dr. Tauber has a long-standing recognition for his generous support of many international projects and research activities, and one of these projects is the establishment of the Tauber Bioinformatics Research Center, which is developing the core technology upon which our platform is based. Dr. Leonid Brodsky is the director of the Tauber Bioinformatics Research Center and is an expert in bioinformatics and biostatistics, has numerous publications on bioinformatic algorithms, and has developed a number of different novel algorithmic approaches that are specific to different types of biomedical data analysis. These have been widely used in basic and translational research in biotechnology, pharmaceutical R&D, agriculture, and basic research. Well, what we will discuss today is also a result of hard work by some of the people mentioned here on the left. Julia Panov, who is an expert bioinformatician, has helped prepare many of the materials we will use. Avi Titievsky, who is a computer scientist and leads the development of the T-Bioinfo platform that we will leverage. Mohit Mazumdar, who is in charge of business development for Pine Biotech. Joshua McKendall, who maintains the educational web portal we will leverage, and Bipsa Biswas, our online community manager and support specialist, who I'm sure that many of you already had the chance to interact with already. Of course, there are many more on our team that I simply do not have time to mention specifically in this short period of time. But it's really a great team that is all committed to making bioinformatics more accessible. Over the past several years, we have conducted a number of programs for thousands of participants, both online and at a variety of university campuses around the world. We have been working with faculty, researchers, and students to build a collection of essential bioinformatics training modules that cover the important omics data types, methods of analysis, and illustrations that demonstrate applications of bioinformatics to various life science domains. So the online Omicslogic programs are built on this foundation. As in all of our programs, this upcoming Omicslogic Data Science program will give you access to all of our online training resources and include special daily meetings for 1.5 hours each day that will provide a hands-on experience and interactive learning sessions that are much different from what you typically get from online courses. The program is a combination of interactive sessions, asynchronous coursework, coding assignments, and support from our team that will be available to assist you and help you overcome any technical challenges that you might face. Over the course of the program, you will get a chance to build a personal profile, completing online coursework, answering quiz questions, completing assignments, and gaining new badges to demonstrate your accomplishment. The point system is based on detailed data that reflects participant activity, including time spent in modules and quizzes, different grades, practical analysis evaluation, and points for completing coding challenges. Upon completing the program requirements of participation and minimum course completion, you receive a certificate of participation that you can always use on your resume, linking to a profile that also shows all of your achievements over time. For those that decide to continue and work on a project of their own for the duration of the program, you will also gain personal feedback from an expert in the field that we will match you with. When the project is complete, you will receive a special certificate of excellence. That's why our previous program participants rank the Omicslogic programs at the top based on program interactivity, personal feedback, support, and the quality of projects that you will be able to achieve leveraging the resources that are made available as a part of this program. So let me briefly speak about the Omicslogic Data Science program. What is it all about, and what are you going to expect to get out of this program? Let's start from the beginning. What is data science, and how is it related to biomedical or omics data? The truth is that data science is a recent term that is used to represent a variety of different things: statistics, computer science, and a variety of different applications in different domains. For example, data science could mean preparing data for analysis when you extract data from financial markets. It could also mean that you're analyzing advertising revenue and different audiences, or preparing sentiment analysis when you analyze social media data. When we talk about biomedical data, that is the domain knowledge that you must have to be able to apply effectively computer science and statistics principles to extract knowledge or inference from the data that you have access to. So when we talk about data science in the context of biology, that is typically or traditionally has been referred to as bioinformatics. Bioinformatics in itself is also a diverse field. Bioinformatics covers collection of data, annotation of data, access to different types of data, as well as the tools to analyze, visualize, and interpret this data. So we are going to focus in this program specifically on the Omicslogic data science portion of bioinformatics. So we'll talk about briefly different types of omics data, formats, and challenges associated with some of the new types of data that are becoming more and more widely accessible. We're going to talk about how to use exploratory data analysis to prepare the data appropriately for the different types of analysis for downstream analysis, and how you can use different types of statistical properties of the data to understand whether you have to normalize it and remove outliers. We'll also cover a topic that has been becoming more and more popular and interesting in a variety of domains called machine learning. Now, machine learning is a term that has been around since the 1950s and has really transformed our ability to deal with big data. Surprisingly, in biomedical data or biological data, new technologies called high throughput technologies have really made the big data problem central to discovery and application of data analysis in this field. So we'll talk about machine learning, different types of machine learning, and discuss the specific methods of analysis and interpretation, because ultimately the goal of this program is to help link the raw sources of data to the different tools and methods of processing, analysis, visualization, and to help make scientific, sound, and logical interpretation of the outputs from these different methods. So in the program, we have combined a number of interactive sessions that we will conduct for an hour and a half each day over the course of 2 weeks, a total of 10 different sessions where you will be able to ask questions. We will introduce topics, and we will also guide you to complete specific self-guided study materials that will expand on the topics that we don't have time to cover in these interactive sessions. In the self-guided study materials, we will suggest some study materials for you to complete before our next session, but we realize that the time to go in depth into each step of analysis will take a lot of time. So we will offer you additional time at the end of the program to complete some of these resources on your own self-guided time. We'll also do practical assignments. These include big data processing pipelines, as well as coding assignments in both R and Python. So let me briefly speak about some of these in greater detail. An important application in biomedical research for machine learning has been in both clinical and pharma and biotech, as well as the practical research or the basic research applications. And so we'll talk about these starting from the general logic, which helps us understand the objective, the problem that is there for us to solve. And then we'll talk about examples where some of the machine learning tools and data science principles have been used effectively to find interesting insights about these problems that touch every one of us. Now, it's important to understand these challenges and problems in industry and both research because they apply to anyone involved in the life science research and application. And when I mean everyone, I mean those that are in the lab, so lab technicians that are generating more and hopefully better data that need to understand how this data is utilized. I'm talking about those that are going to develop new data analysis tools and integration, which is a huge challenge right now in biomedical research, as well as those that have to make decisions based on some of the tools, models, and data sets that are available. So we're really talking about bioinformatics or biology as a data science that is applicable for all of these categories of participants. Let me talk about some of these different topics in greater detail. So Omicslogic Data Science program will start on August 3rd, next week on Monday. Each day, for an hour and a half, we will meet together and we will discuss a variety of topics. These are divided into 2 different weeks. The first week is going to be dedicated to data preparation, understanding the different types of omics data and their properties. These are associated with big data challenges and opportunities, and they require effective cleaning, loading, and processing of data. You also have to know how to explore this data quickly using statistical assessment to understand outliers and normalize your data, preparing it for the specific types of analysis that we will cover in week 2. In the week 2, we will talk about different types of machine learning and artificial intelligence. Now, it's a fascinating topic that we'll touch on a little bit in detail today, but we will talk about it from first a high level, where we have the 2 different categories of supervised and unsupervised machine learning. We'll talk about the models that are available and how to train and fine-tune those models. And we'll also talk about evaluation of their accuracy and how to make them applicable to a more general application or project. And we'll also talk about production quality. So when we talk about production, we're actually trying to solve real challenges. So we'll provide a few examples of how they are applied in the real world. So when we talk about data processing, we're really talking about different data formats that are really changing as a result of a variety of different high throughput omics technologies. And they require a combination of methods that are combined together in a sequential way, forming what we call bioinformatics pipelines. For example, when we talk about different types of omics technologies, we're really talking about technologies like next generation sequencing, mass spectroscopy, different types of structural data. And each type of these data sets could be fine-tuned to study different topics. For example, we can study variation at the genomic level, mutations. We can study methylation, which will allow us to understand the structure of chromatin and how it changes gene expression. We can also talk about proteins that are being expressed and how they interact with each other, protein-protein interactions, or even study the microbiome, the variety of small microorganisms that can shape together as a community our ability to process different types of foods or react to different types of environmental stresses and challenges. So we'll spend the first part of the program talking about these different types of data and how they are processed. How do we turn them from raw data, a raw direct output from that machine, from the technical instrument that produces raw data, into what we call structured data. After that, we will introduce our big data analysis multi-omics platform that you will have access to, where you can analyze these data sets without having to have extreme high performance computing resources on your own. Typically, in a production environment, you would design a pipeline and then apply this pipeline to a variety of different data sets utilizing a predefined space and computational resources. So that's exactly what this platform provides. Now, you will also discover that as you learn about these pipelines, you actually get to understand the logic of this analysis. And the logic of this analysis is reinforced by color-coded maps, explained methods with references to in-depth review in research articles, a variety of input and output formats that allow us to both visualize the data and understand how this data is associated with different phenotypes, and also is a guided process. So it eliminates a lot of the mistakes that people typically make when they first start building those pipelines. Now, as a result, you also get to understand what reproducible workflows really are so that you can take that further and apply that logic to a variety of problems that you will face in the future. Now, behind each one of these methods is a piece of code, a script. And so as we look at these different steps, we'll take a few of those examples and dig deeper to understand how does it actually work and what stands behind each one of these buttons. Then we'll also talk about data visualization and normalization. So an important part of working with these types of data sets is understanding what they represent. And when we say what they represent, because we have so many different indicators, for example, over 20,000 genes or over 3.2 billion base pairs in the human genome, we really have to understand what they speak about, what they can tell us about a variety of different conditions. And so when we do that, we have a number of coding challenges that will help us understand how you can apply best practices in both R and Python to visualize this data and make that visualization more impactful. For example, in our practice examples, you will see how you can select an example of a specific type of a visualization, learn about the code, and as you learn about the code and the outputs, you then get access to try yourself how to go through this code, change some of the settings, and in a step-by-step fashion, develop an understanding of what the code does at each part, and then how does it produce some of these visualizations. So by changing some of this code, and by practicing in different environments, for example, in R and in Python, you will really start understanding what are the benefits of using some of this technology. So we'll also see how in the future, as you use some of these methods, you will be able to work with some of this information inside not only on our Omicslogic code platform, but also as you download the code that you've produced on the platform and then modifying it on your computer so that you can also take that away from the program. We'll also show how some of these apply in biomedical research. So we'll talk about different examples that you can start playing around with, different types of data. For example, for cancer diagnostics and an understanding of cancer biology, or an understanding of single cell data and tissue composition. How you can also study the microbiome and different types of infections at the genomic level or at the diversity level. So ultimately, when we say projects and example data sets, we actually mean taking publications from cutting edge research articles and journals and breaking those down in terms of what the authors did, where did they find the data, what type of data it was, what kinds of methods they applied to that data, and ultimately, what is the use, the practical application of the type of analysis that they performed. When we move on to week 2, we'll start talking about data mining and classification. So first about data mining. When we have big data sets, it's important to understand how these big data sets can be very complex once they are taken as a whole. And we have specific methods that can allow us to identify patterns in these complex data sets by using data dimensionality reduction and clustering. So we'll talk about how when you have your source data, right, and the source data is unlabeled, you just have a number of samples, you can apply a variety of different algorithms to really understand what is in the data in terms of patterns. And these algorithms give us novel information. So this new information that we get out of these algorithms is the association or identification which group they belong to. So we'll talk about a variety of these algorithms, including different types of clustering, principal component analysis, and other types of dimensionality reduction techniques that apply to different types of data differently. These will be both a high-level overview that will provide with clear explanation of why and when and how to use some of these methods and for what purpose, like PCA for data exploration and visualization, and some of the considerations that you need to have and that really make a difference when you try to apply these different types of data sets. When we talk about clustering, we'll actually talk about the specific process of going from bottom up or top down approaches. How do they differ? What kind of information do they provide? And how can we apply them to a variety of these different problems? We'll start from an intuitive, user-friendly interface. Well, all you have to do is upload your data, start your process, and then understand what are the specific settings of each algorithm. So when we talk about these specific settings, we can try to apply different types of settings, change those parameters, and see what are the outputs. But then we also have to learn about the specific linkages, distances, expected counts, and what does it mean that the data is being transposed when we actually input it. So we'll go a little bit deeper to understand how the different aspects of linkage and distance can help us understand what do we actually mean when we deal with data, right? Because this is not going to be distance in a three-dimensional space between objects. This is going to be distance in a multi-dimensional space in a matrix. And so all of these different methods of evaluating linkages and distances will play a role in what we ultimately get as a result. So when we analyze our results, we'll actually look at visual representation and link how the different settings that we put into the method give us different types of outputs. We'll also see the relationship between visualization of one type of data and visualization of the same data in terms of how much information it provides, because the objective of the analysis and matching analysis visualization is to really provide us insights into in-depth biological and biomedical problems. For example, we'll take a look at the Cancer Genome Atlas, that provides us multi-omics data sets about a variety of different types of cancers, where we can start realizing what are the important factors about tissue specialization, as well as cancer processes that we would like to study using some of these analytical tools. Now, from a data science perspective, we'll also see how some of these different types of methods are applicable not only for a output that gives us a new group or an idea about what the data structure is. It is also a very useful tool for data visualization. So we'll see how some of these examples really are great examples to improve our visualization techniques and to interpret the data that we have in a much better way. Now, after we talk about dimensionality reduction and clustering, we'll talk about classification. Machine learning is typically referred to two types of problems: classification or prediction of a specific class based on labeled data, as well as regression. And so regression is a prediction of your next output from a continuous data set. And we won't as much focus on regression, but we will focus on classification. So in classification, we will talk about two specific subsets or subproblems, which is prediction itself and feature selection. Supervised machine learning is a process when we take source data and are able to label that data to prepare a training data set, which is then used to train an algorithm or a machine, a computer, to be able to predict what it learned from the training data set on some test data. Now, the magic of making the training data set representative of a problem that we're actually interested in, and then having the right set of test and validation data is critical to making sure that your model is going to be effective and is going to be able to adapt to unexpected types of challenges that it will face in the real world. So we'll call it a black box for now. But this black box has a name, and each one of these algorithms that we put in the black box will approach the problem a little bit differently. So when we are able to offer a number of different examples, a number of different project data sets that have real world implications about treatment, about diagnostics, about selecting a specific type of an approach to treat a patient, we will see how these different algorithms actually work. And so when we look at the output and we're able to understand the output that this algorithm provides us with, we'll also be able to evaluate both the accuracy and the feature significance of our data set. So these are very useful tools that, in the context of machine learning, provide us with an additional insight into how we can start working with the data sets. These different types of methods are organized by different types of decision trees, discriminant analyses, or support vector machines. And so we'll take at examples. This will not be a comprehensive overview of all of the possible methods out there, but I think it will provide you with a real clear understanding of how different categories of methods actually work and what are some of the important parameters for us to tune when we try to apply these data sets to a specific problem. For example, we'll talk about random forest, and we'll talk about the problem of bagging and applying multiple decision trees to get a good prediction consistently and also do feature selection as we go through that process. We'll talk about how we can take support vector machines and modify the way that the support vector machines deals with classes, also to give us an understanding of how it can be applied to different types of kernel problems, different types of complex classes, and also feature selection in the context of support vector machines. So let me briefly explain what you will do after you learn about the conceptual part of the program and are able to apply some of these data sets, so some of these methods to a real data set. So when I talk about this code platform, I am talking about a specific number of tools that we have designed to support your learning process. So first, when you get to start the program, you will actually register on the platform, register for the program. So let me show you briefly what I mean. On the platform, as I mentioned before, there's a variety of different resources, starting from asynchronous coursework to the sessions that we will have as an interactive session. And once you join this program and register for membership, you will have access to all of these upcoming sessions. In each one of these sessions, we will introduce a particular topic, and you will be able to ask questions and get support through the forum that is monitored daily, and where we can provide you with additional information about all of these different methods and data sets that we are covering. You will also get access to coursework. In the coursework, you will get much more in-depth explanation of the different topics that we cover in the program, and have practical project examples that you can practice on. You will be able to monitor your own progress by looking at what you have completed, where you have left off, and tracking the points that you get as a result of each one of your activities. You will also get personal feedback on the assignments that you submit. Inside the courses, there are assignments that you will complete, and those assignments will be actually evaluated by a person who will provide you with comments, with additional links to resources that you can study, or coursework that you can review to make sure that the concept is clear. Now, some of these concepts are going to be based on coding, and some of these are going to be based on an interactive platform that you can actually use without any coding, right? So we'll kind of go from a high level overview, more and more in-depth and more technical. And depending on how technical you want to get, you can actually get pretty far by downloading eventually the scripts onto your own system and working directly in Python or R to make sure that you understand how all that we do on the platform and we do in the coding exercises could be carried on without these tools. Now, an important part of these data sets is that they are huge. When we're talking about transcriptomic or genomic data, literally every sample has over gigabytes of data. When you talk about such data, having a coding environment is not enough. You have to have a lot of RAM, you have to have a lot of CPU power. So what we are trying to do is to level the field for anyone that does not know coding, that does not have those kinds of resources, to still be able to actually do real research. So when you get access to the platform, you will be able to use first demo pipelines that do not require any kind of inputs and outputs, but just really require for you to understand the logic of the analysis. And once you understand the logic of the analysis, you can see the output immediately without waiting for anything to happen at all. For example, in this case, we are looking at output of a genomic visualization pipeline, where we can see how individual reads from a particular data set are aligned to a reference genome, right? And we can explore our data at the read level. In another example, when we want to do an estimation of gene expression, we can actually build a combination of other types of tools, and those tools will allow us to study how high or low each gene is expressed. And as you can see, when you go through this process, you actually get to understand what each step does and quickly get an output. And we will talk about these outputs, and we'll talk about the different biomedical challenges that these problems are illustrating, how the bioinformatics methods or pipelines applied to this data set help us understand what is in these outputs. Now, at the next level, you will be able to go to the platform and actually create these analyses yourself on a project of your own. Now you will have to make some decisions about what type of data you're putting in and how will that data be processed using the same tools that you've learned about in the coursework and in the demo pipelines. After you run some of these, you will see that some of the results will actually include outputs as downloadable code. So here, for example, I will look at an example pipeline that I ran, and you can see that one of the outputs is a pack in R. I can take this package in R and actually play around with the same code, understanding what it does in these exercises or tutorials, and then playing around with them, again, using a cloud environment. So we are offering two options on this cloud tutorial space. The first option is guided coding tutorials. So let's take a look at an example. Here, we took the same code that was in the output from the pipeline, and we are taking a gene expression data set to understand what can this gene expression data set tell us about the problem that we are studying. Once we understand it, we go through a breakdown of the code. We understand what libraries are used, how do we input the data, what do we actually do to plot the data and to perform the analysis. After that, you get to try it yourself. You actually can change the input data set, you can change the parameters, and you can run this data set to understand what will it actually produce. This runs on our server. So when it runs on our server, you will see an output provided to you just on the browser by running on the server. So for example, here, I got the same result from this principal component analysis in a 3D view. In Python, however, we do not offer a server to run your analysis. In Python, when you select the same exercise in Python, you will be able to actually run this on your own computer. Now you'll see the differences in code and understand how it relates to solving the same type of a challenge, but in a different coding language, but you will actually have to load all of the information in your browser. So the next step, after you have completed your coding challenge, you can download the full code and you can actually go into your own environment and start playing around with that code to make sure that you understand how to set your working environment, your data sets from your computer, and make the output save into files. Now, there are additional benefits to that because you can actually call specific functions that can only work on your computer and not on the server that we provide you. So we hope that this process is going to be effective for all of you to go from a high-level overview of what we are doing in terms of the logic of analysis, to see the different examples of projects, to then to be able to be further into the code itself and eventually set it up on your computer. So let me tell you what are some of the coding exercises that we have prepared for this program. So we will talk about different tools like differential gene expression, which really uses statistical analysis as well as some clustering to make effective visualization of differentially expressed genes. We'll talk about principal component analysis as a dimensionality reduction analysis for these complex data sets like gene expression. We'll talk about clustering and different types of clustering like k-means or hierarchical clustering. And we'll also talk about classification, the problem where we have to assign classes to our data sets and then see what types of methods are giving us the best prediction. Also evaluating what is this prediction quality, how accurate is it, and how stable it is when we apply it to a different validation data set. Finally, we'll talk about a variety of different applications. So I think what's fascinating in terms of data science for biomedical data is that you can quickly see the application of these tools to very important and significant data sets. These are not data sets that are about generating money. These are not data sets that are about social sentiment and interaction between people. These are data sets that really explain the basis of diseases, different biological processes at the subcellular level, and infection using examples from pathogens that we're all painfully aware of, like COVID-19. So we really put a lot of effort into preparing project examples, curated data sets, and specific guidance to give you a background before you jump into your analysis. If you've seen, like me, any other types of examples on data science and machine learning, you know that in each presentation, at least 30 minutes is dedicated to explaining a data set. And unfortunately, we get to use very simple data sets that are actually about value of homes or square footage or petals and different types of plants. So I think it's very important for us to here understand what types of challenges we're going to be solving. So one example that we'll be using is the application of machine learning for prediction of survival for cancer patients over a time frame of about 5 to 10 years. So this is a huge challenge. When you look at a patient today, how can you actually predict, will that patient survive? Will it have a recurrence of the same disease? What are their risk level? What are their chances of survival? And we'll talk about specific ways that we can use this type of information to make decisions about treatment selection. We'll also talk about a novel type of cancer therapy drugs. And these drugs have started to become more and more popular because of their effectiveness, starting from a drug called Herceptin that was introduced in the 1990s, so not so long ago. And we'll talk about the relationship between the biological understanding of how these different diseases occur, develop, and manifest in different patients, and what are the specific types of indications that we can extract from big data that allow us to target those specific pathways. We'll also talk about the application of big data, right? So once we have a collection of big data, how is that actually changing our ability to analyze a new patient that comes in, providing a much more accurate diagnosis by assigning that patient to a particular class or group that we have already previously studied. So we'll actually talk about a number of different examples. Like I mentioned before, these examples are both in the clinical domain and in the pharma and biotechnology domain. So in the clinical domain, we'll talk about examples of patient stratification, how we can group patients by risk, and what do we actually call risk? How do we know what their risk is? Disease classification and diagnostics. So how do we know what a disease is and how do these machine learning tools actually change our understanding of disease and how they actually affect today our ability to diagnose diseases like cancer or like individual types of infections that have resistance to specific treatments or infections that are caused by different pathogens. They are also applied to identification of potential responders to therapy. So we can actually study a patient today and predict what is the most likely course of treatment or combinations of treatments, or what kinds of interactions we should be aware of to make sure that the treatment or the therapy is effective. Obviously, in the basic research domain, the mechanisms of disease onset and progression are critical because ultimately the machine learning model that we designed has to be easy to explain to someone that needs to trust that machine learning model. So a big challenge with machine learning is when the machine learning model predicts something very obvious. If you tell a patient that they should be taking this drug or another, you just have to say that you're a doctor for that patient to trust you somewhat. But if you're talking to doctors that have years of experience and know that this drug and that drug have a similar mechanism of action, or they perform a similar function when they metabolize, you actually have to convince them that the machine learning prediction that this drug should be used instead of a drug that they're familiar with, you actually have to be able to explain the model performance and what features it actually is based on. So that part of it is very critical for machine learning adoption in the clinical world. In pharma and biotechnology, as you all know, it takes over 7 years to bring a drug to market. So there are critical challenges to making sure that the new drugs that are discovered using accumulated data from thousands of patients in a variety of conditions are actually going to be effective, and they're going to be used effectively when they are on the market, so to say. So there's a variety of challenges like biomarker discovery. How do we understand, based on molecular features, which patients to treat with what drug? Detection of toxicity at the in vitro stage. So before we start testing this in humans, how do we actually understand that a drug will not be toxic, right? How do we eliminate the potential of long-term toxicity? For example, side effects that are related to chronic heart conditions when we are trying to, treat mild conditions like inflammation in joints or something like this, right? So how do we detect that at an early stage? Analysis of molecular mechanisms of a drug action or target discovery, and also drug repurposing or repositioning. How do we actually apply these tools to solve challenges when we talk about known approved drugs that are known and studied, potentially to have new indications or new applications to new types of treatments? So I think very, very important and very practical applications that we can try and apply these methods to. Now, I think the exciting part of it is to actually try and apply the methods that you will learn in this program to data sets where you kind of know what the outcome would be at first, right? So at first, you can really see the outputs from your own analysis and compare them to analysis that an expert team has done. And once you know how that matches up, you can actually start applying those to new types of questions. So a particular objective for these programs for us is to be able to allow you to develop a new project idea. And we have a set of guidelines that we have prepared that we think will help you in that journey. So in fact, I think that, and Bibsha, maybe you can share this link also for the participants. We have a guide on how to prepare your own project. So as we talk about tools and the different applications and the different example data sets, how do you actually start developing your own project? So again, this project or this program will consist of 2 weeks. And in those 2 weeks, we will cover a lot of ground to get you into this domain of big data and different types of tools to process, analyze, visualize, and interpret the data sets. After the 2 weeks are over, you will have additional time to work with our support on an independent project or on working on one of the projects that we have prepared as an example for you to learn from. Now, I want to conclude this with some registration links that you can use to complete your registration if you're interested in this program. We have a process where you first fill out a pre-registration link, and please put these links on the chat if possible. So the pre-registration link, and let me actually go to this link, will get you to a page where you learn about the program topics. You can also ask us questions, obviously, in the session. After I complete, we'll have some time, about 5 minutes, to answer any questions you might have. And once you fill out this information, you send it to us, and our team actually reviews this information and sends you some additional information on what to do if you would like to register, who is this for, and what is the best way for you to use this program, right? So we want to make sure that it's very clear this program is designed specifically for those that are interested in big data, specifically in omics data applied to biomedical domain, and would like to go beyond kind of an interactive high-level bioinformatics. So they would like to get some coding experience as a result of this. But it is not a coding workshop. This is not a program to get you to be proficient in R or Python. We hope that you are either already familiar with one of these languages or are committed enough to independently pursue additional help with getting proficient in those languages. We will simply not have enough time to do that in this program. So, but you will get some experience, you will get an understanding of the different methods, and you'll be able to interact with our team to get help if you decide to go deeper into one of these topics. Okay, so after you do this, you actually you receive a link to complete your registration on the main program page. Now, once you complete this registration, you create a profile for yourself. This profile is where you start accumulating points. So I'll explain what this means. You can see here an example profile of one of our participants. You can see that once they register, and by the way, the registration itself is free, and we also have some free tools available for you. You will see that a person who has registered will have to create a profile where all of their activity is accessible so that we can track and address any issues that come up. You will have a point system that helps you estimate how far along you are in this process of learning. And you can also continue picking up from different courses that you have started before, continuing to make progress in those courses and completing those. Now, each independent course is either included in the program or accessible for free, or some of the courses might not be a part of this program. So you'll have to see which courses are actually accessible to you in the program. And once you complete them, you get individual certificates for completing those courses. And they will also be available right here under your profile. After you do that, you can continue to go deeper into one of the projects that we make accessible. So those projects will illustrate some of those applications that I've already covered. So that program is going to give you a personal profile. That personal profile helps you start building some of that points, some of those points, and eventually the top participants are actually highlighted on our leaderboard. And so when we highlight those participants on the leaderboard, we also tell their story. We also explain the projects that they have done. And I believe, Bipsa, you have a link to some of those projects that have previously been done by some of our participants. We're very encouraged and proud to have received, a lot of very high quality feedback for these programs. And so we think that, if the fit is right for you and you are ready to commit the time and, the resources in terms of how much effort you will have to put in to actually make the most out of these programs, you can get a lot from these different programs. So I'm going to pause now, and I'm just going to make this open for any questions that you might have. I'm going to, ask Bipsa and Mohit to let me know if there are any questions also on YouTube, and I'll be able to continue in addressing those questions in the future.
Thank you, Ilya. I think we have addressed the questions on YouTube already. So I would like to ask if anyone has a question now, we can address those as well.
Okay, and also I have a few questions here that are unfortunately just on Zoom. Okay, so there's a question. I'm a beginner with no prior knowledge of data science. Still, I am able to understand it, but it would be better if you can explain some technical terms for beginners. Yeah, so that would be the exact objective of this program, is to provide you with an overview of both the high-level logic and the specific terms that are commonly used. We'll use that both as a general session explanation of what these terms are, but also we'll see an example. So let me actually show you an example in a publication, right? So one of the objectives for this is for you to then be able to read a publication and really understand what they mean. So here you can see how we used least square support vector machines and random forest algorithms to identify molecular features associated with responses of a collection of 70 breast cancer cell lines to 90 experimental or approved therapeutic agents. So you can see how right here in the abstract, which is just a few sentences long, you already have to know what is least square support vector machines, what is random forest algorithm, what are molecular features, how do we find or establish association, how do we measure responses, what are these breast cancer cell lines, right? So a lot of questions. So we will go into detail about each one of these terms. Okay, so how much coding do you need to know? The coding that you can start with zero coding experience. Literally, if you have zero coding experience and you start, you will quickly understand how some of the methods are done using coding. Now, you can also see, as a result of this program, what type of languages are there and what kinds of languages are best for what type of application and what are some of the considerations, right? So hopefully, if you don't know any coding, you'll be able to decide whether you want to pursue more specialized training in R or in Python, right? Or you want to just do a little bit of both, right? So that is one of the outcomes that we hope to achieve from this program. How long live sessions? The live sessions are going to be 1.5 hours long. So they will include an overview of a topic, a practical exercise that we will go through together and make sure that you're able to do that exercise, and then have some time for discussion. So if you have any questions, we're able to address those. Okay, Professor Babu, you're an vivo pharmacologist, interest to take up the course, but I have knowledge on statistics, but beginner for data science and no knowledge on coding or computer programming. I think that's a perfect program for you because it does require for you to kind of understand logically what's going on, but then with the computer programming or coding, you will get guided tutorials at first, then you'll download them on your computer. We'll actually provide you with some tools where you can start practicing. For software, really will guide you, and maybe I'll give you an example here. If you go, well, eventually once you're registered, you'll see here a clear explanation of R and Python and how to set it up on your computer. So it's a real interactive tutorial that you can go through. And if you have any questions about this, you'll really see what we'll help you kind of install all of this. And basically, the only things that you will need is to install R and Python on your computer. RStudio is preferable just to give you an environment to work with. And then we'll go through some of these in here, which is an online platform. You don't need anything to run these examples. And we'll go from very basic on how to load, manipulate and visualize your data. And it's going to start with very simple exercises. Okay, so hopefully I answered that question. What special alterations apps? So again, not really any apps. All you would need is RStudio and Python to be installed on your computer. Okay, great. Well, thank you for attending today. Hopefully, Bipsa and Mohit, if you could post some of those links again for those that are interested to review some of the benefits of the program. Again, I think the program is unique in the sense of online courses. It is not a typical Udemy or Coursera course where you just watch videos and then do something on your own and hope to get some feedback. This is a very guided program, and this program is going to be, really focused on the outcome. So it is actually in our interest to get you to a place where you can demonstrate your profile and the projects that you have prepared as a real validation of what you have learned. Right, so you can see some of the examples here and some of the feedback from real participants. We also have a page on some of the project outcomes. Bipsa, if you have some of those links, we have a number of recent projects that people have prepared in our past programs, and we run these programs all the time. So there's really a variety of topics that come up in different programs that we conduct. Okay, so thank you again for joining this webinar. We're going to be continuing to reach out to you with these links by email so that you have them after this webinar is over. And we welcome all of you to join come Monday on August 3rd. So we still have a few days for any one of you that is interested to complete your registration. Please reach out to marketing@pine.bio if you have any additional questions about the registration process. Thank you for joining, and we'll see you again soon.